Okay, so after doing the big stuff, let's do the small stuff. So we're going to focus now on drawing the skeletal muscle fiber, the myofibril, and the sarcomere. I'm going to draw the fiber kind of small so that I can make my myofibril and my sarcomere much bigger. I'm thinking, it's fine. It's been just a little bit of time since I've actually drawn this out. But what we're going to do is maybe draw a cylinder like this. And I'm not going to draw like the whole cell. I'm just going to show you what would be inside of that cell. And I guess I should kind of draw it all around like that, duh. All right, so we're going to think about this, and what we're going to do is we're going to put a little bit of blue on the outside edge right here, and this is the endomysium. Just so you know, the endomysium is going to cover the top part of the muscle fiber so that you understand that this red line is going to be the sarcolemma. The sarcolemma is going to be the cell membrane just with a new name, and it's given a new name mostly because it's a charged membrane. So this is going to be negatively charged, is negatively charged, negatively charged on the inside when it's at rest all the way along, okay? The sarcolemma is going to have little openings inside of it that dip into the cell just like this. And I'm only going to draw some of them at the top, but you want to think about the sarcolemma dips into the cell and goes transverse through the cell all the way to the other side of the cell. These are called the transverse tubules. Sometimes they're abbreviated just as the T tubules. Okay, just like that. The transverse tubule is then going to be linked to a modified smooth endoplasmic reticulum. So it looks like this kind of netting that goes from one T tubule to another T tubule, just like that, right? You have to forgive me when I draw the muscle fiber, it's very difficult to draw it like on a whiteboard or in a white background like this, and it be understandable. Like this is something that's three-dimensional that I have a hard time really replicating in 2D. Okay. So we have a nice netting that goes between our transverse tubules like that. Actually, that doesn't look so bad. Kind of made it look a little bit okay. So there'll be more sarcoplasmic reticulum all the way along right here. So remember I said that's a modified smooth endoplasmic reticulum. This is called our sarcoplasmic reticulum. It is often abbreviated as the SR, okay? I don't abbreviate it on the test. So you should know sarcoplasmic reticulum. Then also inside of the cell, you're going to have, a bunch of mitochondria. You'll have nucleus. You'll have a nucleus like, nuclei like all over the place, bulging out there. The really most important thing is that, remember, this has cylinders on the inside of it. So we're going to have these other cylinders, right, like this. And actually, what I'm going to do is I'm going to erase this and draw this a little bit bigger, just because I don't want to be stingy with my size here. So these cylinders make up probably 80 to 90% of the contents inside of the skeletal muscle cell itself, okay? Just imagine that I keep drawing my T tubule, I'll draw one down here to not obscure my picture. Geez, that got out of hand. So my T tubule is going to come all the way down like that, And then my sarcoplasmic reticulum, just remember your sarcoplasmic reticulum goes around the myofibrils. Okay, so just recall or remember that. If you don't want to draw all this a sarcoplasmic reticulum in, then just don't draw it in. I just want you to see that it's going to go around my myofibrils. So this is the myofibril. This is a specialized organelle that's only found in skeletal muscle and cardiac muscle, and it's going to be made up of proteins. It's made up of myofilaments. Those myofilaments are proteins called actin and myosin, okay? So the smaller unit of the myofibril is actually going to be the sarcomere. The sarcomere is the contractile unit of the muscle, and it is going to span from a Z-disc protein to a Z-disc protein. So what you're going to see is, now I have a hard time drawing this, again, round and three-dimensionally, so I'm just going to draw it flat to make my life easier. So we're going to see this chevron pattern is going to create our Z-disc, and I'm going to do my ultimate best to do this properly, but I never make my Z-discs even. I've just never been able to do it, okay? So we're going to do a Z-disc and a Z-disc, and then we do another Z-disc over here, like that. And then maybe, you know, you have another one underneath your sarcoplasmic reticulum. So you want to understand your myofibril, your myofibril is made up of individual sarcomeres that are all connected together. So we have our Z-disc. Right here, a Z-disc and a Z-disc. Okay, so from Z-disc to Z-disc is the sarcomere. So there's another sarcomere. Just like that. In the middle of the sarcomere, I'm going to make one protein, and this is called the M-line protein. The M-lined protein is going to help us with making sure that some of our filaments, our myofibrils, stay in place. In particular, you're going to have two types of myofilaments. If I said myofibrils, I'm sorry, it's very late. It's been a very long day. We're going to have two types of myofilaments, okay? One myofilament is thick and one myofilament is thin. So our thin myofilament is actually composed of three different types of proteins, and the thin ones are going to connect to the Z-disc, just like that. So the actin is the purple, and I'll draw this a little bit bigger down here, but the actin is going to kind of be a little bit rounder, and then it has an underside that has a depression in it, like that. And then this is the myosin binding site. So you have your myosin binding sites on what's called the actin protein, okay? The other part, excuse me, the other part of the thin filament is you'll have this wavy protein like that. I'm not going to draw all of them, but this wavy protein is going to cover the myosin binding site, and this is called tropomyosin. Okay, you're thinking, oh, that's in the myofibril, the way that I drew it like that. No, just erase your myofibril right there. Okay, and then we're going to see that we're going to have another protein that holds the tropomyosin in place, and this is called troponin. Okay, so on here, you're going to have like little yellow dots, and that makes up our thin filament. So the thin filament is going to be held in place by the Z-disc. The thick filament is going to be between the thin filaments here and is actually going to be held in place by the M line, just like that. And our thick filament, yes, is kind of like this line, but we want to see that it's going to have all these little rounded heads that come off of it. So I usually just kind of draw it like that, with these like golf club like heads that are going to be sticking up and sticking down like that. So you can kind of think about like those heads go like little balls like that. I drew it kind of small. I wish I had drawn it a little bit bigger, but this is myosin with the myosin heads. Okay, just like that. What you should see on the sarcomere is when you have these overlapping proteins, it's going to create a banding pattern. So it's going to create an A band and an I band. And in order to see this, you kind of have to make at least 2 sarcomeres. So I'll make another one over here. And, you know, forgive me a little bit if my drawing is off and not perfect. I've never really been able to perfectly draw a sarcomere, even after all the years of me drawing it on a whiteboard for students. See, I just didn't draw it correctly. It's slanted a little bit because I didn't draw my Z-disc correct. That's what you start to realize after you've drawn this a couple times. But that's fine. It looks crappy, but you'll get the picture. I hope so anyway. So the banding pattern that we're going to see is that we're going to, I don't know what colors to use. I'll use different colors. So at the end of the actin, right about there, and the end of our actin right about there, what we should notice is we have a little bit, actually, I'm sorry, I have to redo this. It's not the end of the actin that I'm, so sad. I have to draw that again. That happens sometimes with erasers. It should be fine. It's not the end of the world. So I'm going to redraw this. It's not the end of your actin, I'm sorry. It's the end of your myosin. At the end of your myosin, what you should see is you have a little bit of overlap between actin and myosin here. And the overlapping area between actin and myosin is called the A band. Just like that. Because it's a darker banding pattern. It's going to not have a lot of light go through it. That's why it's called the dark band. And then you're going to see that from the A band all the way over to the end of another myosin right there, this is the I band. This is the light band. So this is where we have only the thin filaments and the Z disc overlapping. So I want you to kind of think about this in terms of a bigger picture. And what I'm going to do is I'm going to erase this part down here, just so you can see what I mean when I say like all of the I bands and all of the A bands come together to help us create the striated pattern of the muscle. So if you did still have your myofibril right here, like this, what you're going to see essentially is here is where our A band is. Then we'll have an I band. Then we'll have an A band. Then we'll have an I band. Then we're going to have an A band. Then we'll have an I band. And you see how this A band, which I'm just going to totally mess up my drawing altogether. See how those A bands all line up perfectly and all the I bands line up perfectly. So in the muscle fiber, this is what's creating that dark area, the light area, the dark area, and the light area, like that. Okay. So we're going to draw the sarcomere, or I might have it drawn for you. I don't know. I'll probably draw the sarcomere one more time when we look at muscle contraction. Okay. So the next part is going to look at the physiology of the muscle contraction.